Hello, everyone, and welcome to today's live broadcast on control strategies and method development for nitrosamines in APIs and drug products. I'm Shannon Stoltz for LCGC and pharmteco, and I'll be your moderator for today's event. We are pleased to bring you this webcast presented by LCGC and Pharmaceutical Technology and sponsored by SK pharmteco. SK pharmteco is an analytical testing and global contract development and manufacturing organization specialized in the production of APIs, advanced intermediates, and cell and gene therapies. SK pharmteco is owned by SK Incorporated, the strategic investment arm of South Korea's SK Group. Now, before we get started, we do have a few quick announcements. First is that this webcast is designed to be interactive, and we encourage you to ask questions throughout the live event. You can submit questions by typing them into the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, and the slides will advance automatically throughout today's event. If you have any technical problems either viewing or hearing this presentation, please click on the question mark help widget in the top right of your presentation window. Moving on, I would now like to introduce our speaker for today. We are very pleased to be joined today by Shankar Sankaran. As Director of Analytical at SK pharmteco, Shankar oversees method development, validation, release testing, stability studies, and reference standard management for both CDMO and contract analytical services. Prior to taking on this leadership role, he worked as a laboratory chemist, where he supported API programs from early development to commercial stages. And before I pass it off to Shankar, we do have a poll question for the audience today that should be appearing on your screen soon. What is the most critical factor when selecting a partner to outsource your analytical testing? So if the audience could please review the options and select a response, we would greatly appreciate that. Again, what is the most critical factor when selecting a partner to outsource your analytical testing? And we'll just give everyone a chance to answer that. Okay, hopefully that gave everyone enough time. So as you're answering that, I just want to thank everyone for participating in the poll. And Shankar, thank you so much for being here today again. And if you could please get us started.
Thank you for the kind introduction. So as today, we'll talk about control strategies and method development for nitrosamines in APIs and drug products. We'll go through a handful of regulatory requirements and what those entail, kind of what are the technologies that are used for nitrosamines testing, how do we establish and validate methods for screening and for routine analysis of nitrosamines, what are the challenges associated with nitrosamines method development, strategies for establishing test methods for drug-derived nitrosamines or NDSRIs, and then a couple of case studies diving into NDSRIs specifically. Really, this applies really for kind of most late phase drugs. You're completing a risk assessment. You're in the process of getting your NDA, or if it's for already marketed drug, you're completing a risk assessment for nitrosamines to include as part of your filing. And as part of these assessments, when there's a potential risk for the presence of nitroso impurities, whether that's the typical small molecule nitrosamines or a drug-derived NDSRI nitrosamine, you'll be required to put a method in place. And so let's talk through what putting a method in place looks like. Right. So just taking a step back from a specification and really kind of establishing limit of quantitation for methods, you need to understand what limits you're going to be required to control to. So regulatory agencies across the board, the FDA, EMA, and so on, have kind of established acceptable intake limits for nitrosamines that are ever expanding. As they gather more data on product-specific nitrosamines and NDSRIs, those are being added to the corresponding regulatory agencies' websites. But in addition to that, both the FDA and EMA have harmonized on a risk scored-based assessment of nitrosamine limits. And so they have specific guidance using a CPCA scoring mechanism to establish these limits to get to a stage where you calculate the acceptable intake limit, and based on that, set specifications for your corresponding drug substance or drug product based on dose. For some of the most challenging nitrosamines, NDEA, NDBA, those limits are very low, right? Because the acceptable intake limits are down to 26.5 nanograms per day. That for an equivalent dosage of 200 mgs, you're talking about 0.13 ppm or 130 ppb. And the larger your dose, of course, the lower it goes, right? So if you have a drug dose that's 1,500 mgs total per day, then you're really looking at 20 ppb for NDEA. And that's where you start to run into challenges with development and limits of quantitation. For product-specific nitrosamines, NDSRIs, right, that can range even lower because the EMA has some language about setting limits as low as 18 nanograms per day when there's no determinable score or determinable acceptable limit. And there can be, or it can go relatively high as well, right, up to 1,500 nanograms per day. And so you have to have a wide-ranging method, and you have to have a method that can really go down to low limits of quantitation. So we'll talk about how people approach this, right? So for generally, people are using mass spec, of course, for this approach, and you need sensitive methods with limits of quantitation in the parts per billion to meet the low acceptable intakes recommended for nitrosamines. There are established methods published by the FDA and many others, of course, and USP that can be good starting points for developing methods. But you need a target LOQ that's 10% of the limit. And really, you're setting 10% of the limit to justify kind of no routine testing. If you can demonstrate across batches that you're consistently below 10% of the proposed limit, then you can ultimately have enough data to waive putting it on your specification long term. If you set your LOQ to 30% of your limit and have methods in place that can quantify down to 30% of your limit, you can justify skip lot testing. So once in every 5, 10, 20 batches, depending on your risk assessment. Testing is typically achieved through GCMS or LC-MS-MS. There's different strengths and weaknesses to both that I'll talk about. Primarily, you're going to see a lot more methods that are LC-MS-MS focused. The best approach to this that I've seen, and the approach we use in-house, for example, is setting a couple of platform methods in place. So we have a platform method on our GCMS system, a platform method with our LC triple quad to complete a quick screening and then go into a more product-specific matrix development. As far as NDSRIs are concerned and drug-derived nitrosamines are concerned, high-res mass spec is the ideal application. You know what specific mass you're looking for. The higher the resolution mass spec you can get, the more sensitive you can make your methods for those applications. Okay. So from a platform method perspective, having a typical platform method in place makes the screening process go a lot faster, right? So really, once you have the established chromatography conditions in place, what you're optimizing for is your sample matrix. And the sample matrix is what presents the most challenge. Maybe less so for a drug substance, but certainly more so for a drug product where you have to deal with the insoluble excipients. A platform GCMS method is best for the smaller nitrosamines, and it can be a fast option. Typically, there's less work to optimize for the sample matrix, so you can move pretty quickly. The cons are really that it's hard to get a truly sensitive method with just GC single quad. You can have a little more success with a GC triple quad, but your limits of detection, limits of quantitation will be higher than the more robust LCMS or LCMS-MS methods. The other downside to GCMS specifically is the potential for in situ nitrosamines, depending on the sample matrix, because you'll be typically using headspace or even potentially within the GC inlet. High temperature conditions can potentially generate in situ nitrosamines depending on the nature of your drug. So in addition to GCMS, having a platform LC-MS-MS method is suitable and can be used for a wider complement of nitrosamines. There's a method that we've got that's established across two orthogonal columns. The reason for that is if you have depending on the nature of your drug, the nature of impurities within your drug, you may have coelution concerns that impact specificity or recovery of specific nitrosamines. And so having an orthogonal method in place that you can quickly jump and screen to is useful. The development here takes a little bit longer. You're usually looking at 2 to 4 weeks. And the primary reason for that is matrix-related interference. You're trying to control for matrix-related interference, trying to find the optimal solubility, extraction conditions for your nitrosamines. And then from there, you can take that platform method and validate it within 2 to 3 weeks. The advantage to LCMS and LCMS-MS is it's easier to optimize for sensitivity and specificity. You can really narrow mass ranges, narrow collection times, increase ionization capabilities, both at the source and downstream with MSMS. And so your sample concentrations can be lower. The cons are the sample matrix issues are a bigger challenge than the typical GCMS method. You would need to figure out that there's a ton of solubility concerns, a ton of interference concerns that you have to work your way through. But having a couple of orthogonal methods can help speed up that process. Then, as you can see, really the detection and quantitation ranges can go a lot lower, right? You can go down to the nanograms per mil and then range your sample size based on your limits, right? So we've taken this LCMS-MS application and driven it down to where you could quantitate to single parts per billion and even less than 1 part per billion, depending on the needs of both the nitroso impurity and the specific drug in question. So the typical validation for an LCMS-MS method looks like this, where we've established an LOQ down to 1 or 1.5 nanograms per mil, and then you can scale your sample preparation based on that. Recoveries are range from 85-90% to about 140%. You're looking for acceptable recovery that's in the range of 50% to 150%, and precision of not more than 25%. And with these methods, you can be relatively precise. Solution stability is a big concern as part of these validations. The typical standards have been stable for about 4 days. And then depending on the nature of your sample, it can be anywhere from 24 hours or less, sometimes up to 3 days. All right, so we'll dive into a couple of case studies here. First, we took a short nanopeptide, bradykinin, as a sort of proof of concept for how you would implement a control strategy for nitrosamines and peptides. Initially, we completed a screening using the orthogonal methods on the LC triple quad. Spiked recoveries from the sample matrix were expected for all screened nitrosamines except 2, and you can see the 2 in red on the column on the left. For NDEA and an NMBA, using this hypersil method, we were having low recovery for one and extremely high recovery for other. The NDEA recovery issues were determined to be a coelution issue with the API. And so the advantage of having an orthogonal method, you throw it on the orthogonal method, you create a different set of separation conditions between your API and NDEA, and your recovery looks good for NDEA jumping to that. For NMBA, we continue to struggle with recovery, with over-recovery. And this seems to be an issue with the matrix or the diluent, a combination of the matrix and the diluent. And so effectively, what happens is all injections after the samples, including the bracketing standards, we're showing a 2x response for NMBA specifically across the board. That was in samples, that was in standards. And so something about the sample introduction process and sample matrix it specifically causes a recovery issue with NMBA. And so this is one where we're still working on it, trying to solve a challenge where specifically we're really evaluating the sample prep and the diluent additives to make sure that we can create a robust method for the recovery NMBA. Our second case study was for a method that was developed simultaneously for a drug substance and a drug product for the 4 smaller small molecule nitrosamines, NDMA, NDEA, NDIPA, and NEIPA. At first, we applied our routine LCMS method. The challenge with the LCMS method is the solubility for the drug product was poor, and that's primarily due to the excipients present in the drug. The API, as it by itself, achieves complete dissolution in the diluent, in the selected diluent. It's an aqueous diluent formic acid. We completed a slew of different slurry extractions from the excipients. Those extraction mechanisms were efficient with good recovery for the nitrosamines. We did this in the absence of the API. However, when you introduce the API with in that process, it co-elutes with multiple nitrosamines, impacting the recovery. And so across the board, you can see recoveries are very poor for that LCMS method. We try to our various orthogonal LCMS methods and continue to be an issue. Co-elution of the API with the target molecules continues to be an issue. Sample matrix related interferences continue to. For this specific application, we switched over to the GCMS method, where you can remove a lot of the components that cause matrix interference. And so dissolved diluent acted into DCM matrix-related interference issues were resolved, and you can see good recoveries across the board on both the drugs, for the drug substance and for the drug product, for all 4 impurities. Okay. Next case study involves a drug substance and a drug product, and this is a gel capsule drug product, so the excipient profile is a little bit different. Initial screenings on the GCMS looked promising. Solubility was good for both the API and the drug product. However, in the GCMS results, we were looking at, we observed elevated levels of NDMA. And due to the nature of this drug specifically, we suspected that the elevated levels of NDMA were being formed in situ, rather than innately present in the drug substance or the drug product. And so we completed a solvent screening where we're looking at the presence of different diluent matrices, and effectively what we saw is diluent you exhibit confirmed our observed levels of or observation that in situ formation is what we're seeing with the presence of NDMA. And so, however, needed for solubility of the drug itself. And so we took an aqueous diluent, basified it with potassium hydroxide, and used that as the final diluent. And so that base diluent provides the best stability for NDMA quantitation. Other aqueous diluents weren't stable and couldn't be held for more than 30 minutes. And so once you put that basified diluent in place, we had stability for up to 24 hours, and then had a robust method in place that could help drive the completion of that method and validation of that method. Switching gears a little bit to NDSRI control. So nitrosamine drug substance-related impurities are structurally similar to your intermediates or APIs most of the time. You need high-quality reference materials of these impurities in order to get good sensitivity and specificity. And the biggest challenge that you're going to see is that because they're so closely related in structure, getting chromatographic separation can be a challenge. And so generally you need sensitive methods in the parts per billion quantitation, usually in the tens or hundreds of parts per billion quantitation limits, to meet the low acceptable intake. This is usually done via high-res mass spec, a QTOF, an Orbitrap, something of that nature. Some NDSRIs are theoretical. You can propose them based on the presence of secondary amines, sometimes tertiary amines, but we've struggled to synthesize them in the lab. And so there are a couple of approaches that I'll talk about that to address the theoretical presence of impurities as well. Just as a reminder, the FDA has got published guidance that was updated in the beginning of September of 2024 that recommends the conclusion of NDSRI confirmatory testing of drug products by August 1st, 2025. So keep in mind that for drugs where you're actively in the NDA process or already marketed drugs, you need to be following these requirements. So jumping into case studies for NDSRIs, here we're looking for a nitroso API and a tableted drug product, with the biggest challenge being matrix again, right? So the tablets don't dissolve in common diluents. We had to complete an extraction in a methanol formate buffer mixture. And the next challenge is the chromatography. The NDSRI co-elutes with the API, and so all development efforts that we could put in place for screening stationary phases, mobile phases, etc., failed to get baseline. We couldn't baseline resolve the API from the nitroso impurity. And so here we're leveraging the strength of a high-res mass spec system where we're leveraging the mass resolution to quantify the nitroso API. So you create a really narrow mass filter for the API, for the nitroso impurity in question, and sort of establish your specificity that way. The challenge with that is that you need to have a very clean instrument and a good tune for accurate analysis. Extraction efficiency was demonstrated through increasing volume studies. So we want to make sure that if we're going through setting an extraction process rather than a complete dissolution process, we have a demonstrated recovery of the NDSRI. And with a 1x and 2x volume extraction process, we demonstrated that the recoveries are relatively accurate for what we're expecting. Okay, so when you have a risk assessment that suggests a potential nitrosamine, there are a few approaches to demonstrate compliance and control. One of these is a protocol issued by the WHO for a nitrosation assay procedure. And it's a general in vitro assessment for the potential formation of nitrosamines. So we've completed this for a handful of drug substances and drug products. And HUNAP studies are really kind of specifically meant to subject your drug to nitrosating conditions to determine the formation of possible formation of nitroso impurities. Specifically, this reference in the EMA guidance, as well as a couple of Q&As on the FDA for nitrosamines, suggests that it's recommended HUNAP studies be conducted on starting materials, intermediates, and APIs during the manufacturing process to better understand the potential formation of nitrosamines or lack of formation of nitrosamines. And then if you have positive findings, then carry that further to figure out an acceptable intake limit, whether that's through the CPCA risk scoring or through formal toxicology testing, such as Ames testing. Okay. So the process for the HUNAP study is you set up the API and the nitrosating conditions, you scan for the mass range surrounding the proposed nitroso impurities, and then you fine tune with a narrow mass range or a single reaction monitoring process to get adequate quantitation. And so here in this case study, we took a 10 millimolar API solution to ensure material is completely in solution. You dissolve it in low pH with organic solvents as needed. You mix it with this 40 millimeter nitrite solution that's maintained at around a pH of 3 to 4. And then you maintain the reaction mixture at 37 C. And so this is the WHO procedure. And you maintain that at about 4 hours. And so you create optimal conditions for nitrization. And then we completed an initial screen, run a Q1 scan for a wide mass range, identify potential matches with the proposed nitroso impurities. You extract these product ions further and run an SRM with fragmentation to confirm structure, because you have the potential to generate impurities that match the mass, but ultimately aren't the nitroso impurity of interest. And so once you've confirmed the generation of the nitroso impurity, then you further develop this method and refine it. You can use this procedure to generate your reference standard through fraction collection and purification, and then develop a quantitation method to go along with it. Okay. Next case study was for a single theoretical NDSRIs and a small molecule API. So in this process, in the synthesis of an active pharmaceutical ingredient, the formation of a diazo group is accomplished through reaction of an amine with a nitrous acid to generate this diazo compound. In this reaction step, there's a potential to form, it goes through the nitroso impurity process before it forms this diazo. A pH-controlled extraction procedure was developed for sample preparation. Then you use a LC high-res mass spec. We use our LC QTOF to develop and identify and quantify the proposed nitroso impurities. And so we're screening API samples for the presence of these theoretical nitrosamines at the masses suggested for each of these proposed nitroso impurities. The impurities, because we're unable to generate a true reference standard for any of these nitroso impurities, the nitroso impurities are unstable, they form the diazo right away. We're screening for a theoretical mass and using a surrogate standard with similar ionization potential to provide a pseudo-quantitative approach to screen for these nitroso impurities. And so these impurities were quantified against this surrogate compound, and this method was applied to analyze 5 representative samples of the drug substance. And so from that study, we were able to develop a method that was specific, linear, precise, with a robust quantitation limit required for those specific proposed NDSRIs. Using that structural analog intermediate as a surrogate, we established a quantitation limit at 2.5 ppm. to meet the acceptable intake limits for a drug product. This is a relatively low daily dosage drug, so your limits are a little bit higher. The four potential nitroso impurities weren't detected in representative batches. So we're screening for this mass, we're screening for a precise mass for each of these nitroso impurities, and ultimately could not find a response related to these impurities in our high-res mass spec screening. And so there were peaks observed at these masses that needed further refinement to demonstrate that there weren't the nitroso impurities. We evaluated the mass spec against the proposed nitroso structures, right? And the primary masses observed in each scan region was an intensity of 4 orders of magnitude larger than the equivalent nitrosamine mass response. And so we recognize that the specific responses are being derived from the product itself rather than the specific nitroso impurities. And so we've taken this approach, provided an LC-MS screening of proposed theoretical NDSRIs, provided the data using a surrogate quantitation approach, and successfully completed filings with this approach. In the next case study, there were 3 theoretical NDSRIs that were proposed as part of the drug substance. There's concerns on specificity of the nitrosamines with the API, so evaluation was completed with the original purity method as well as an orthogonal method. And so, as I've noted previously, specificity for the for NDSRI specifically can be a challenge because they're so structurally similar that chromatographic separation can be a challenge. So here we're looking at the response of the specific mass for each proposed nitroso impurity. And then the method for determination for trace nitroso impurities was shown to have sufficient sensitivity using a surrogate compound with similar ionization potential. And so we're using this surrogate to demonstrate a proposed limit of 0.01 ppm, and the method sensitivity was confirmed using this prior stage surrogate. Representative crude API and pure API materials were analyzed, and the potential nitroso impurities were observed. And so here, for a handful of these proposed nitroso impurities, we did see a response in the API, and so further evaluation was needed. So these impurity peaks were submitted for fragmentation to evaluate the presence of the typical nitroso fragments, but those fragments weren't observed. And so fragmentations for nitroso impurities are expected to show a loss of that NO radical or a loss of this amino nitro radical. And so that's a documented phenomenon, and we're using that to demonstrate and justify that the proposed impurities or the impurity response that we were seeing in this LC-MS work isn't actually a nitroso impurity. It's another impurity that's related to the API. And so we observed, in addition to the demonstration that the nitroso impurities, the observed response for these impurities wasn't the nitroso impurities, there was also a screening completed to demonstrate that whatever these impurities were, looking at it in the crude API versus its purging capability as you take it to the pure API process, you don't see able to purge these impurities of interest. So even if there was a potential concern that these were nitroso impurities, there's a demonstration that the production process can purge these impurities as you go through the purification crystallization process for your pure API. So either way, there's a control mechanism in place for these impurities. So that's the typical approach for nitrosamines and nitroso impurity control. Nitrosamines guidance from the FDA and EMA, they present a significant analytical challenge. You need to have very low LOQs. You have to wrestle with sample matrix issues, sensitivity issues, solubility issues, and so on. And so requiring a highly sensitive and robust method can only be achieved via mass spec techniques, GCMS, high-res mass spec, LC-MS, MS. Platform methods can be useful for typical nitrosamines and can be established based on the current FDA and USP methods. Once you have an accurate and sensitive method, chromatography development is minimal. The challenge is developing for your individual sample matrix, whether that's your drug substance and solubility issues or your drug product and solubility and interference issues. And so structural similarities to impurities will also play a factor here. Having orthogonal separations kind of pre-developed and in place can help assist in rapid screening for impurities and accelerate the development and validation process. And when you jump to NDSRIs, you almost always need high-res mass spec or LC-MS/MS as a primary testing strategy. Accurate mass with narrow mass filters can be a tool for especially challenging impurities, especially where you can't get chromatographic separation. WHO and app studies can help provide supportive data on the formation of theoretical nitrosamines, and then you can take that further and isolate reference materials as needed for further characterization and development of methods. And then single reaction monitoring and fragmentation provide clarity on the identity of impurity peaks observed with masses consistent with the NDSRI. So if you get a response for a proposed nitroso impurity that matches up with your NDSRI, you can carry that further with fragmentation to demonstrate the presence or absence of those nitro leaving groups and nitro radical leaving group, or demonstrate that you don't see that, and the absence of that demonstrates that it's a impurity related to the API, but not the nitroso impurity that we're proposing. Okay. I want to acknowledge a few people that helped pull all the content together for this and all the lab experiments to go around this. Our team leads and our analytical scientists across the SK pharmteco network have helped drive all of the content that you've seen today, and just a big shout out to them. With that, thank you. I'll thank you for the time. I'll hand it back over to Shannon to get started with the Q&A.
All right, great. Thank you so much for that informative presentation. And before we dive into that Q&A session, I would like to quickly remind our audience how to submit questions. You can submit any questions you have by typing them into the Q&A box, which can be found below the video player. And our first question for you today is, at what clinical phase would nitrosamines testing requirements apply?
So that's a good question. Typically, you would want to be tracking these early in your development process. So what we recommend is really from phase 2 onwards, you have an eye on what you're going to do as a nitrosamines control strategy. Practically, the completion of the risk assessment needs to be done along with at the time that you're submitting your NDA. So really, it's when you're gearing up for a registration, and then commercial manufacturers when you absolutely have the control strategy in place. But especially knowing that the raw materials, the chemistry, the equipment train potentially all have an impact on potential formation of nitrosamines, it's something you want to be keeping an eye on early in the clinical development.
Great, thank you. On to our next question. Is testing required if a risk assessment doesn't identify any potential source of formation of nitrosamines?
In general, the answer to that is no. You don't typically need testing if your risk assessment doesn't identify a specific source. I will say your risk assessment has to be really sound and really robust. You have to do a deep dive into all of the inputs for your materials, all of the raw materials, reagents, solvents, et cetera. Within the guidance specifically, the FDA flags that, hey, even if it's not a typical solvent where you would expect to see secondary amines, for example, something like toluene, where you wouldn't expect to see secondary amines, but they've noted in the past that there's been the presence of secondary amines, and so as a result required some level of control and testing or demonstration of data that demonstrates the absence of nitrosamines. So your risk assessment, as long as it's very thorough in its evaluation of each of the inputs, each of the manufacturing components, container closure components, excipients, et cetera, you wouldn't generally require nitrosamines testing.
Thank you. And the audience would also like to know, is there an easy way to reference previously calculated acceptable intake limits or example limits for other drugs?
The FDA actually has a published website where it talks through both for impurity limits that have been established via actual mutagenicity testing, as well as for impurities where they've made calculations based on the carcinogenic potency categorization approach. You can go into and reference on the FDA's website these values and these numbers to look at, okay, if I have if I'm working on a drug that's previously on the market, and or if I have a drug that's structurally similar to something on the market, can I go in and take a look at these acceptable intake limits and make a determination based on the available data? We've in general, you're going to want to present your scoring, your carcinogenic potency categorization scoring for yourself within your own risk assessments. But the FDA does provide an easy reference for previously established limits.
Perfect, thank you. With that, it does look like it's already time to wrap up. Oh, wait, actually, we have one more question. If the risk assessment is not finding any NDSRI, but FDA has listed theoretically possible NDSRIs on their website, how should one proceed?
So this is a really good question, something we've wrestled with as a challenge over the past couple of years. In one of my case studies that I presented, right, actually in two of the case studies that we presented, at both instances, there's an approach where if there is a proposed NDSRI, and especially within synthetic labs haven't been able to actually synthesize this NDSRI, what we've done is taken a surrogate impurity and completed a scanning, a scan via high-res mass spec for these established quantitation limits based on the surrogate, and provide demonstrable data that, hey, we're looking for this specific mass of this NDSRI. We have an estimated quantitation capability based on the surrogate impurity, and demonstrate the presence or absence of this NDSRI.
Great, thank you. And with that, now it is time to wrap up. So I want to start by thanking the audience for attending and participating in today's event, as well as our sponsor, SK pharmteco, for making today's webcast possible. We would like to ask everyone in the audience to participate in a brief survey that will be appearing on your screen after today's presentation has ended. And you will also receive an email alerting you when this webcast will be available for replay. And we invite you to forward that announcement to any of your colleagues who may have missed today's live event. Otherwise, we hope to see everyone next time. Thank you and take care.